Introduction. A genetic renaissance unfolds. As we stand at the threshold of 2028, humanity finds itself at the precipice of a genetic revolution that would have seemed like science fiction merely a decade ago. The convergence of technological advancement, scientific discovery, and growing clinical confidence has ushered in an era where understanding and manipulating the human genome is no longer a distant dream, but an increasingly commonplace reality. From curing previously incurable diseases to preventing genetic disorders before birth, the next era of human genetics promises to fundamentally reshape medicine, healthcare, and our understanding of what it means to be human. The journey to this moment has been extraordinary. The Human Genome Project, completed in 2003, gave us the instruction manual for human life. In the years that followed, scientists learned to read this manual with increasing proficiency. Now, in 2028, they have begun to edit it with remarkable precision. This article explores the revolutionary developments that characterize this pivotal era, the applications that are saving lives, the ethical questions that loom large, and the possibilities that stretch into a future we are only beginning to imagine. The gene editing revolution, beyond CRISPR-Cas. When Jennifer Doudna and Emmanuelle Charpentier shared the Nobel Prize in Chemistry in 2020 for their development of CRISPR-Cas9, it seemed the pinnacle of gene editing technology had been reached. Yet the years that followed have revealed that they disrupt other genes. The delivery to the right cells and the right tissues remains challenging, and for many genetic diseases, a single gene modification isn't sufficient. They require the precise rewriting of multiple genes or complex regulatory sequences. The innovations of the last 5 years have largely addressed these shortcomings. Prime editing, developed by David Liu's team at the Broad Institute, has emerged as a transformative refinement. Unlike CRISPR-Cas9, which cuts both strands of DNA and relies on the cell's error-prone repair machinery, prime editing works more like a molecular word processor. It uses a modified Cas9 protein fused to a reverse transcriptase enzyme, allowing scientists to insert, delete, or correct DNA bases with remarkable precision. By 2028, prime editing has moved from laboratory demonstrations to early clinical trials, showing promise in treating hemophilia, sickle cell disease, and beta thalassemia with fewer off-target effects than traditional CRISPR approaches. Base editing, another innovation that has matured significantly, uses modified Cas9 enzymes fused to deaminases, proteins that convert one DNA base into another. This technology has proven particularly valuable for treating genetic diseases caused by point mutations, those single-letter typos in the genetic code that can have catastrophic consequences. In 2028, base-edited therapies for hereditary retinal dystrophy have shown remarkable results in clinical trials, with patients experiencing restoration of vision after years of progressive blindness. The delivery of these gene-editing tools remains one of the field's greatest challenges and triumphs. Viral vectors, particularly adeno-associated viruses, AAVs, and lentiviruses, have been refined to improve their specificity and reduce immune responses. Non-viral delivery methods, including lipid nanoparticles similar to those used in mRNA vaccines, have been adapted for DNA delivery, opening possibilities for treating diseases in tissues previously thought inaccessible. By 2028, several phase 3 clinical trials using these optimized delivery systems are showing success rates that would have been unimaginable just 5 years earlier. Personalized medicine, the age of genomic precision. Perhaps the most transformative aspect of the next era of human genetics is its application to personalized medicine. As the cost of genome sequencing has plummeted, a full genome can now be sequenced for under $100, and as our understanding of how genetic variations influence disease susceptibility and drug response has deepened, medicine itself is being reimagined. In 2028, the concept of a one-size-fits-all medication is increasingly antiquated. Pharmacogenomics, the study of how genes affect medication response, has moved from niche interest to standard clinical practice. Patients presenting with conditions like hypertension or depression routinely undergo genetic testing to determine which medications they're most likely to respond to, and at what doses. Insurance companies now cover these tests routinely, recognizing that the cost of genomic testing is offset by improved outcomes and reduced adverse drug reactions. Cancer treatment has been revolutionized by advances in understanding tumor genetics. In 2028, cancer is increasingly being treated not as a disease defined by its location in the body, breast cancer, lung cancer, pancreatic cancer, but by its genetic signature. Tumors with specific genetic mutations now have targeted therapies, dramatically improving survival rates. A patient's cancer might be sequenced within days of diagnosis, and treatment decisions can be made based on the specific genetic drivers of their particular tumor. Immunotherapies, refined through a better understanding of how tumor genetics interact with immune system genetics, have expanded the arsenal of effective treatments and improved outcomes dramatically. Polygenic risk scores, which synthesize information from hundreds of thousands of genetic variants to estimate an individual's predisposition to complex diseases, have matured into practical clinical tools. Someone identified as having a high genetic risk for coronary artery disease can now receive tailored prevention strategies beginning in their teens or 20s. Similarly, women with high polygenic risk scores for breast cancer can benefit from earlier screening or preventive therapies. While these scores are still imperfect predictors, genetics is only part of the story. Environment, lifestyle, and luck play crucial roles. They provide a quantified risk assessment that allows for truly personalized medicine. The pharmaceutical industry has been transformed by advances in understanding genetic variation in drug metabolism. Drugs are increasingly being developed with companion diagnostics that identify the patients most likely to benefit. In 2028, it's rare to see a new drug approved without information about genetic biomarkers that predict response. This has led to paradoxically smaller patient populations for individual drugs, but with much higher response rates and fewer wasted treatments on patients unlikely to benefit. Preventive genetics, stopping disease before it starts. One of the most profound implications of advances in human genetics is the possibility of preventing genetic diseases before symptoms ever manifest. By 2028, this has moved from theoretical to practical reality in several domains. Pre-implantation genetic testing, PGT, used in conjunction with in vitro fertilization, allows prospective parents to select embryos free from severe genetic diseases. What began with screening for the most severe conditions, such as Huntington's disease or cystic fibrosis, has expanded significantly. Genetic counselors in 2028 must grapple with increasingly nuanced questions about which genetic variants warrant embryo selection, especially for conditions with variable penetrance or late onset. The practice, while still relatively rare and expensive, has become less controversial as its safety profile has been established and as more families have exercised reproductive autonomy. Newborn screening programs have expanded dramatically. In 2028, most developed countries screen for 50 or more genetic and metabolic disorders at birth. Earlies. Identification allows for early intervention, transforming the prognosis for conditions that would previously have been detected only after irreversible damage had occurred. Infants with severe combined immunodeficiency, once a death sentence, can now be identified before symptoms and treated with gene therapy, allowing them to develop normally. For adults, the ability to identify genetic predispositions to disease has enabled a shift from reactive medicine to proactive prevention. An individual identified as carrying a pathogenic variant in a BRCA gene, which significantly increases breast and ovarian cancer risk, can undergo enhanced surveillance or preventive mastectomy. Someone identified as having familial hypercholesterolemia can begin treatment early to prevent premature heart disease. These interventions, while often challenging from a psychological perspective, save lives. Somatic gene therapy, rewriting the rules of disease. While germline gene therapy, modification that affects all cells and is passed to future generations, remains one of the most ethically contentious areas of genetics, somatic gene therapy, which affects only the body's non-reproductive cells, has matured into a realistic therapeutic option by 2028. The first somatic gene therapy approved by the FDA was in 1990 for adenosine deaminase deficiency. For decades, approvals trickled in slowly. By 2028, there are over 24 approved somatic gene therapies, with many more in clinical trials. These therapies are treating a diverse range of conditions: hemophilia, spinal muscular atrophy, inherited retinal diseases, and severe combined immunodeficiency, among others. Hemophilia, a bleeding disorder caused by mutations in genes encoding clotting factors, has been transformed by gene therapy. Patients who required frequent infusions of clotting factor, with all the attendant risks of infection and inconvenience, can now receive a one-time gene therapy treatment. By 2028, many hemophilia B patients are now effectively cured, with their bodies producing adequate clotting factors without ongoing treatment. Perhaps even more remarkable are gene therapies for blindness. Inherited retinal dystrophies that lead to progressive vision loss and blindness have long been among the most devastating genetic disorders. In 2028, several gene therapy approaches are restoring vision to patients who have been blind for years. These patients describe the experience as miraculous, as they regain the ability to navigate their homes independently, recognize faces, and see colors again. The challenge with many somatic gene therapies is one of delivery. The eye is relatively accessible. Delivering gene therapy to every cell in the liver or brain is far more challenging. Yet innovations in delivery are making the seemingly impossible achievable. Engineered viruses, optimized for systemic delivery and designed to evade immune detection, are enabling treatment of diseases affecting multiple organ systems. Non-viral delivery methods, particularly those using lipid nanoparticles, are being refined to improve cell targeting and reduce off-target effects. The ethical frontier, navigating the moral landscape. As the capabilities of human genetics have expanded, so too have the ethical questions they raise. By 2028, the field has moved beyond the theoretical debates of earlier decades to confront real, practical dilemmas that require thoughtful navigation. The distinction between therapy and enhancement remains central to ethical discussions. There is broad consensus that using genetic technologies to treat or prevent disease is ethically justified. The challenge arises at the margins. If we can use genetic technologies to treat severe myopia, nearsightedness, can we use them to optimize vision? If we can treat hearing loss, can we create superhuman hearing? What about genetic variations that influence height, intelligence, or athletic ability? These are no longer abstract philosophical questions, but practical ones that clinical geneticists and bioethicists must address. Social responsibility initiatives. Yet significant gaps remain, particularly between wealthy nations and developing countries. Informed consent in the era of genetic testing raises complex challenges. As the cost of genetic testing has fallen and the speed of technology has accelerated, the psychological and informational burden on individuals has grown. People might be tested for genetic variants of uncertain significance or for predispositions that may never manifest as disease. The proliferation of direct-to-consumer genetic testing has democratized access to genetic information, but has also created a landscape where people may misinterpret results or receive information without adequate genetic counseling. The use of genetic information by third parties, insurers, employers, governments, remains a significant concern. While many jurisdictions have enacted protections against genetic discrimination, the regulatory landscape remains inconsistent and concerns about misuse persist. By 2028, there is heightened awareness of these issues, but gaps in protection remain, particularly around genetic discrimination in life insurance and long-term care insurance. Perhaps most contentious is the question of germline editing. While the scientific community has largely reached consensus coordination and of ensuring that advances in genetic technology benefit all of humanity, not just the wealthy nations that can afford them, the frontiers, what lies ahead. As 2028 unfolds, the next frontier of human genetics begins to emerge. The possibilities are both exciting and sobering, stretching toward futures we are only beginning to imagine. Gene drives, genetic technologies that rapidly spread genetic modifications through populations of organisms, hold promise for combating vector-borne diseases like malaria by rendering mosquitoes incapable of transmitting the disease. By 2028, these reproductive freedom, potentially allowing anyone, regardless of sex or fertility status, to have genetically related children. It also raises the intersection of artificial intelligence and human genetics is creating new possibilities. Machine learning algorithms are being trained on vast genomic datasets to identify patterns that human researchers might miss. By 2028, AI-assisted identification of novel disease genes has become routine, dramatically accelerating the pace of genetic discovery. These algorithms can predict protein function from DNA sequence, model the effects of genetic variants, and identify promising drug targets with increasing accuracy. Conclusion: a new chapter in human history. As we navigate the next era of human genetics, we stand at an inflection point in human history. The technologies we've developed and refined have given us unprecedented power to understand and manipulate the genetic basis of human life. We can now prevent diseases, treat conditions that were once inevitably fatal, and envision genetic futures that would have seemed impossible just years ago. Yet with this power comes profound responsibility. The decisions we make in 2028 about how to develop, regulate, and deploy genetic technologies will shape not just the present, but the generations to come. We must ensure that these advances are guided by careful ethical reflection, that they are deployed equitably across all populations, that they enhance human flourishing rather than exacerbating existing inequities. The next era of human genetics is not a predetermined destination, but a landscape of choices. How we navigate it with wisdom, humility, and a commitment to both innovation and equity will define this generation's contribution to human progress. As we look to the future, we do so with both the excitement of possibility and the sobering awareness of responsibility that comes with such transformative power. The scientific achievements are remarkable and will continue to accelerate. But the ethical, regulatory, and equitable deployment of these technologies will ultimately determine whether this era becomes remembered as one of humanity's greatest triumphs or as a cautionary tale about unchecked technological power. The choice, in many ways, remains ours to make.